Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing an exam review for Biology 1408 Exam 2. This exam covers chapters 4, 5, and 6. So let's get started. Chapter 4, touching on cell structures. And I want you to know the difference between prokaryotic and eukaryotic cells, and that the prokaryotic cells are comprised of the domains bacteria and archaea, whereas the eukaryotic cells, they make up the domain eukarya. The domain is called eukarya, and these include the protists, the fungi, the animals, and the plants. One thing you should know is, besides the domains that I just mentioned, prokaryotic versus eukaryotic, you should also know about these basic features of all cells. So I'll be asking about the basic features of all cells. So all cells have at least one chromosome. They've got DNA. In prokaryotes, the DNA is in the form of a nucleoid, and in eukaryotes, the DNA is inside of a nucleus. All cells have a plasma membrane, all cells have a cytoplasm, and all cells have ribosomes. So please know that all cells could possess all of these features here. Next thing to know, prokaryotes. What's special about those? We need to know that prokaryotes lack a nucleus. And what else do they lack? Remember, they lack membrane-bound organelles. I want you to know that prokaryotes lack membrane-bound organelles. And eukaryotes have membrane-bound organelles. They possess a nucleus, and they possess membrane-bound organelles. Now, this is a typical animal cell picture. I'm not going to have anything which requires you to label parts of a cell like this. The exam is made up of multiple choice and true-false questions, and usually one short answer question at the end, but there's no matching or labeling type of scenario. But what I do want you to know in this animal cell is that animal cells possess centrioles. You see this structure here, the centriole? I want you to know that animal cells possess centrioles, but I want you to know that plant cells do not. You see, the plant cell does not have centrioles. Now, I want you to know that there are three structures that plant cells have that animal cells don't. So a plant cell, which is another type of eukaryotic cell, like an animal cell, a plant cell has this large central vacuole in the center. So plant cells have a central vacuole. They also possess this green organelle here. These are the chloroplasts for photosynthesis. And they possess a cell wall. I want you to know that the plants possess cell wall. Okay, great. Let's move along here. What else do we want to know? Ribosomes. I want you to know what the function of ribosomes are. Remember that ribosomes are the cell's protein synthesis machinery. This means that proteins are synthesized by the ribosomes. Ribosomes make proteins. I definitely need you to know that. Also, we talked about the mitochondria and the chloroplasts. I need you to know that the mitochondria is the site of cellular respiration. This is where ATP is made. So please know that mitochondria is the site of cellular respiration, which means the process of making sugar, right? Or, oh, sorry, making ATP from burning sugar. This is, we're going to be discussing cellular respiration in chapter 7, I want to say. Chapter 7 covers the cellular respiration. And I want you to know that in plants, chloroplasts are the site of photosynthesis, right? So chloroplasts, photosynthesis. This is the process of making sugars from using sunlight energy. That happens in plants, in the chloroplasts. By the way, chapter 8, we're going to go into a lot of detail about the chloroplasts. What else do we want to know about? Okay. Now we talked about the ER, the endoplasmic reticulum. Please know about the endoplasmic reticulum. There's smooth ER, smooth endoplasmic reticulum, and rough ER, rough endoplasmic reticulum. What do I need you to know? I need you to know that the smooth endoplasmic reticulum, what are the jobs of the smooth endoplasmic reticulum? The smooth endoplasmic reticulum is where certain lipids are made, lipids are synthesized, certain carbohydrates are metabolized, detoxification of drugs and poisons occurs, and it's a store of calcium ions. So I want you to know that the smooth endoplasmic reticulum is involved in these 4 processes. And what about the rough ER? The rough ER is the place where proteins are synthesized. So the rough ER is a location in the cell where proteins are synthesized. I want you to know that. And what about the Golgi? What about the Golgi apparatus? I want you to know about the Golgi. I want you to know that the Golgi apparatus modifies proteins from the ER and then ships those proteins to their destination after modifying them. So it's the shipping and processing center of the cell. So please know about the function of the Golgi. What else do we want to know? Lysosomes. I want you to know the function of the lysosome. The lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. So it's the digestive organelle of the cell. All right. With regard to the cytoskeleton, let's see. We can actually skip the cytoskeletal material for this exam. So go ahead and skip the cytoskeletal material and the extracellular matrix material for the exam. I hope you got something out of it for learning the concepts. Hopefully it was interesting to you, but I won't be testing you on this material here. So let's move on to chapter 5. Those are the concepts that I want you to know so you can be successful and do well on exam 2 for Biology 1408. Those topics that I touched on in this video. So let's hop on to chapter 5 so that we can highlight some of the most important concepts for exam 2 for you for chapter 5. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead with chapter 5 with our exam review of the structure and function of plasma membranes. So one thing we need to know about plasma membranes is that their job is to be a selectively permeable barrier. Selective permeability is the main job of the plasma membrane. Selectively permeable. We should also know the structure of a phospholipid. Remember, phospholipids make up the plasma membrane. And you should know that a phospholipid, if you remember from chapter 3, is made up of a glycerol and 2 fatty acid tails and a phosphate group. So please know the structure of a phospholipid. This is the structure of a phospholipid. Remember that membranes, like the plasma membrane, must be fluid to work properly. And that cholesterol is a steroid hormone that our cells have that allows the membrane to remain fluid. Therefore, cholesterol serves as a fluidity buffer. That means that cholesterol keeps the fluidity of our membranes so our membranes don't freeze up. So you see here, with the help of cholesterol, membranes stay fluid. Another way of staying fluid is to have unsaturated fatty acid tails. So they're, but what I mainly want you to know is that the job of cholesterol. So what does cholesterol do? It's a fluidity buffer, meaning cholesterol keeps your membranes fluid. That keeps your membranes from freezing up, and that's the job of cholesterol in your cells. What else do we want to know? Do you remember these transmembrane proteins we touched on in the lecture videos? I want you to know the different jobs of the membrane proteins. So some membrane proteins are transporters. They could serve as enzymes. They could serve as cell surface receptors. They could serve as cell surface identity markers, cell-to-cell adhesion molecules, and they could serve to attach to the cytoskeleton. So please know the 6 major functions of membrane proteins. These are the 6 major functions of those membrane proteins. Next, what do we need to know? Oh, I may ask you about the permeability of the lipid bilayer, of the membrane. Remember that hydrophobic molecules, such as hydrocarbons like methane, CH4, oxygen, O2, that's oxygen gas, and CO2, these hydrophobic or nonpolar molecules can pass through the membrane easily and rapidly. So you should know that nonpolar or hydrophobic molecules readily cross the membrane, whereas polar molecules like water, right? Polar molecules or ions, remember, these have a positive charge or a negative charge. These ions, these cannot cross the membrane without help. And do you remember, though, that help is in the form of transport proteins? You see here, channels, carriers. These are transmembrane transporters. So again, polar or charged ions require transporters to cross the lipid bilayer, to cross the membrane, whereas nonpolar molecules like methane, oxygen, and CO2 can easily pass through the membrane Here, like they could pass through the membrane without the help of a transporter. Next, we talked about diffusion. Please know the definition of diffusion. Diffusion is the spontaneous movement of molecules from high concentration to low. And if diffusion happens across a membrane, that's called passive transport. So please know the definition of diffusion and know that passive transport is simply diffusion across a membrane. Does passive transport require any energy? No, because diffusion is spontaneous. It does not require energy. Great. So here you can see the diffusion of these molecules of dye across a membrane from high concentration on the left to low concentration on the right. This is an example of dialysis. Now, how is this different from osmosis? Please know the definition of osmosis. Osmosis is the diffusion of water across a membrane. Osmosis is the diffusion of water across a membrane. So not solute. So here, solute is these molecules that are dissolved in the water. See, the solute is the substance that's dissolved in the solvent. Do you remember these terms? Solvent is the water. The molecules of dye are the solute. When the solute is diffusing across a membrane, that's passive transport. That's passive transport in the form of dialysis. If water is crossing the membrane, that's passive transport. That's diffusion of water. This is osmosis. So again, osmosis is the diffusion of water across a membrane. So please know that. And do you remember how osmosis can affect a cell? These are called tonicity effects. In an isotonic solution, and this is probably the trickiest part of the whole chapter, so you should know. In an isotonic solution, the cell does not gain nor lose water. There's no net gain or net loss of water into the cell, because the solute concentration inside of the cell is the same as outside of the cell. But in a hypotonic environment, water will rush into the cell, causing the cell to lyse. Why? Because you have a more pure water outside of the cell. You have less solute concentration outside of the cell. So what do I want you to know? I want you to know that in a hypotonic environment, water rushes into the cell and the cell lyses, which means breaks open. And this is because the solute concentration inside of the cell is higher than outside of the cell. So water rushes into the cell. Conversely, with hypertonic environments, water rushes out of the cell and the cell becomes all shriveled up, which is called a crenated cell. And why is that? Well, this is because the solute concentration, the saltiness outside of the cell, is higher than inside of the cell. So water diffuses out. Water diffuses from a high concentration of water to a low concentration of water. Okay, so please know, again, isotonic, the cell does not net gain or lose water. Hypotonic, the cell gains water until it explodes, until it lyses. Hypertonic environments, these are like very salty environments, water will net leave the cell and the cell will become shriveled or crenated. Okay, the same thing kind of happens in plants. In an isotonic solution in plants, the water is neither net gained or lost. In a hypotonic solution, water is net gained by the cell, but the cell will not lyse because there's a cell wall. the cell just becomes high pressure. It's called the turgid cell. Conversely, in a hypertonic environment, you should know that water net leaves the cell, and this results in a plasmolyzed or dehydrated cell. Awesome. All right, what else do I need you to know for the exam? This is probably one of the two last things I need you to know, and that is that active transport, active transport. So we talked about tonicity, and lastly, active transport. Active transport is when substances are moving across the membrane against the concentration gradient, and this is going to require ATP. So for example, if there's already a bunch of these dots outside, these spheres outside, and you want to pump more of these spheres where they're already high concentration, notice how you're going against the concentration gradient. This will require ATP because this is going against the concentration gradient. You're going against diffusion. So what you should know is active transport is going against diffusion across a membrane. And that's going to require ATP. It's going to require energy to occur. So this is just a rundown of everything you need to know about passive transport on the left and active transport on the right. See, on the left, these purple dots are diffusing across the membrane. How could that be possible, that these purple dots are going right across the membrane? Do you remember what kind of substances can cross the membrane directly like this, rapidly? Remember, those are small, nonpolar, or hydrophobic molecules. These purple dots have to be small, nonpolar molecules. That's the only way they're crossing the membrane directly. However, these triangles and these blue dots need to be helped across the membrane, and the reason for that, remember, is because they are either polar molecules like water, or they are ions like K plus or Cl minus, right? So they are ions. They will need the help of transporters, protein transporters, or protein carriers. This is called facilitated diffusion because without this facilitator, this transmembrane protein, the diffusion would not have happened. All right, that's perfect. So the rest of this chapter is not required. You can skip that. But the things I touched on in this review are fair game for the 1408 Exam 2. So let's take another little quick break with Gizmo and Wicket. We'll come back with the final chapter and the things I need you to know to do well on the exam for chapter 6. We'll be right back. Welcome back from break time with Gizmo and Wicket. Let's carry on with our final chapter of this exam, exam number 2. That's chapter 6, covering metabolism. What do we need to know? We need to know the definition of metabolism. What is metabolism? Remember, metabolism is all of the chemical reactions inside of an organism. So be able to define metabolism and know the difference between anabolic versus catabolic metabolism. Remember, in anabolic metabolism, small molecules are assembled into large ones, and that requires the use of energy, whereas in catabolic reactions, it's the converse. Large molecules are broken down into smaller ones, and this releases energy. So definitely know that, and then know the difference between these types of energy. So know the definition of energy here. So what is energy? And then know the different kinds of energy. What's the difference between kinetic energy, heat, potential energy, chemical energy? So please know those definitions there. Then know the definition of thermodynamics. What is thermodynamics? And what are organisms? Are organisms considered open thermodynamic systems, closed thermodynamic systems, or isolated? Well, they are open. Organisms are open thermodynamic systems. Remember that from the lecture. or open thermodynamic systems. Then we want to know the first law of thermodynamics. And remember, the first law of thermodynamics, energy cannot be created nor can it be destroyed. Okay, so this is the principle of conservation of energy. Energy cannot be created nor destroyed. So first law versus the second law. The second law of thermo, and that is that with every energy transfer or transformation, the entropy or the disorder of the universe increases. All right, what else do we want to know? We want to know that solids have the least disorder. They're considered the least disordered, followed by liquids. Liquids have more disorder, and gases have the most disorder. Okay, so when you're dealing with the phases of matter, solids have the least disorder and gases have the most. What is a spontaneous process? What is a spontaneous process? Remember, that means a process that occurs without energy input. So a spontaneous process occurs without energy input. It can happen slowly or quickly. I want you to know this. Spontaneous process does not have to occur quickly. A spontaneous process can happen quickly or slowly. So remember that a spontaneous process can occur quickly or slowly. Like, for example, rusting is spontaneous, but it could take years to rust. Next, we talked about free energy. Do you remember the units for free energy? G. Remember capital G for Gibbs free energy. All right. Now, we don't need to know the equation for Gibbs free energy for this class, 1408, so I'm going to skip that. But what I do want you to know is that if a reaction has a negative delta G, a negative change in Gibbs free energy, that is a spontaneous reaction. It's also called an exergonic reaction. If a reaction has a positive delta G, it is a non-spontaneous or endergonic process. Okay, so again, recap. A reaction with a negative delta G is considered an exergonic process, which means it's spontaneous, whereas a positive delta G reaction is an endergonic process, which means non-spontaneous. One more thing you need to know is that negative delta G reactions release energy, and positive delta G reactions absorb energy. All right, what else? So you can see here, again, in an exergonic reaction, the reactants, what you start out with, has a higher G value, and the products have a lower G value. This is a negative change in G, or negative delta G. This is an exergonic reaction. It releases energy. Conversely, an endergonic reaction absorbs energy. It requires energy, and it has a positive change in G. Great. Again, let's skip to enzymes. What do I need you to know about enzymes? I want you to know that an enzyme speeds up a reaction. Enzymes speed up a reaction. That's what an enzyme does. And that means that enzymes are catalysts. Catalysts speed up a reaction without being consumed by the reaction. Okay. What else do I want you to know? I want you to know that... Actually, that's good for chapter 6. We don't need to go into too much detail. But I want you to know, again, that enzymes are catalysts. Enzymes speed up reactions. That's about it. Those are good concepts to know for exam number 2 of Biology 1408. Please note those concepts. I've written them down as we've gone along, so I'm going to make sure to address those for the exam. Please know those concepts in detail, and I think you'll be just fine for exam number 2. I hope you're all doing well out there, and I will see you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.